Welcome to Barry's in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
All right, welcome everybody. Welcome back to In the Interim. I am joined today by somebody you're all familiar with, Dr. Kert Viele, and he helps me co-host some of In the Interim broadcasts. Today, we are going to talk about time travel. Maybe not quite time travel, but statistical time travel. We are going to talk about the time machine and its relevance to platform trials. We'll sneak in a little sports, all in this. So welcome back to In the Interim, Kert.
Thank you very much. Good to see you, Scott.
Good to see you. All right, so the topic of the time machine, and we'll call it the time machine. We have called it that, and we'll sort of describe why. We'll describe the relevance of this topic and why it amazingly has lots of people that get very flustered or frustrated by it. And we'll talk about that. So let's introduce the time machine.
Well, so let's go back in time. Speaking of that too, because you've been working on this since the 90s, something like that, when all's said and done.
Yeah, so let's go back in time. And it's interesting how this, I remember, and many of you have heard some of the episodes we do about sports and drug developers in sports. And as a kid, my father, Don, we were talking about the old pub fodder is the best term I have for this. Sports enthusiasts will argue endlessly about the different talent level of players at different eras in sports. And if you watch the MLB All-Star game, which was very recent here, they did a really nice look back at Hank Aaron hitting his 715th home run, and he beat Babe Ruth in the total, not the career home run record, and it was a big deal at the time. Of course, they played at very different times, even though Hank Aaron played for an incredibly long time. Their careers never overlapped, and so questions about who's a better home run hitter. And you could have those questions about today. Who's a better home run hitter? And Don said something to me that stuck a little bit. He said, if you take two players like Babe Ruth and Hank Aaron, they never played together, but they played with players who played together. So back in the 90s, this wanted to put this into actual algorithms. And so this fantastic thing in sports where Babe Ruth never played with the great home run hitters of today, Aaron Judge, two New York Yankee, wonderful home run hitters. They never played together, but Babe Ruth's career overlapped Lou Gehrig's career, who overlapped Jimmy Fox's career, overlapped Ted Williams' career. Ted Williams' career overlaps Mickey Mantle, overlaps probably Hank Aaron at that point, who played for quite a long time. So there's this bridging, if you will, of Babe Ruth to Hank Aaron and even to Aaron Judge today. So they never played at the same time, but this overlap, you could estimate the relative home run ability of Babe Ruth to Jimmy Fox, and Jimmy Fox to Ted Williams, and Ted Williams to Mickey Mantle, and Mickey Mantle to Hank Aaron. You could estimate all these players by simultaneously estimating the effects of age. The effects of era. Age is a different thing we'll come to, but the effect of the era in which they played. And era matters in home runs. There was a dead ball era, the different effect. Pitchers varied over time. Era matters in which you played, but you can always estimate the relative skill of players that played at different times. So I was involved in an effort with Pat Larkey and Shane Reese, where we wrote a paper that we did exactly this, and we did it in baseball. We did it in scoring in the NHL and golf. And in many ways, golf is probably the simplest of those because there's only one goal in golf. There's really only one stat that matters. How many strokes did it take to play 18 holes? And you have exactly the same setup if you wanted to compare Tiger Woods to Jack Nicklaus or Scotty Scheffler to Jack Nicklaus or Ben Hogan, Tom Watson. You have this incredible overlap where they played in the same tournament at the same time. In the NHL, we have the same thing where they played in the game at the same time, and you can estimate relative performance of players bridging across this. And then we estimated this relative efficacy, and we estimated the effects of error, putting everybody, by adjusting by error, putting everybody on exactly the same scale and estimating, comparing these apples to apples on exactly the same scale. So you could compare Hank Aaron to Babe Ruth. You could move Hank Aaron from 1973 to 1927 and say, what would he have done? Now, we called it a time machine because we weren't interested in, suppose Hank Aaron would have grown up in 1920. Now, he wouldn't have played in the major leagues because he was black and he couldn't have played. Suppose it was the nutrition of the time and grew up at that. We weren't interested in that. We were interested in a time machine. Moving Jack Nicklaus to 2025 as a 25-year-old superstar, what would he do compared to Scotty Scheffler now or Tiger Woods in 2001? Moving them in time. So that was the paper and that was the effort. By the way, that was published in Journal of the American Statistical Association. It was actually picked as the applications and case studies paper to present at the joint statistical meetings, that paper, that effort. So that was the introduction to a statistical time machine, Kurt.
So who did you say was the best?
Which everybody wants to argue about all of that. And so fantastic. Interestingly, in baseball, we did home runs and batting average within that. And we did this in the mid-1990s. And if I remember, it's been a while, I think Mark McGwire was the best home run hitter of all time in that. The best golfer of all time at that point, I believe, was still Jack Nicklaus, but that was pre-Tiger Woods.
Right.
Recent updates of that, Tiger Woods is the best within that. The NHL was fantastic in terms of scoring because there's amazing things. When Wayne Gretzky played, he played in an era where their goal scoring was much higher. He was still the greatest scorer of all time, but it's not nearly what I expected. His numbers dwarf everybody's, but he played in an era. And so these are all the fantastic things to talk about. By the way, as we start to move this to clinical trials, and we'll figure out why would anybody care about a time machine in clinical trials? Seems like such an odd thing. It's much more challenging in sports because players over that time are at different ages. And the age of the participant certainly matters within that. Jack Nicklaus at 55 was not Jack Nicklaus at 30. So we had to model how players age within the different sports, and it's different in golf and baseball and hockey. So there's a component of that which adds to challenges of that. And go to that paper and you can certainly read about all of that. But that's different than clinical trials.
All right, so let's move to clinical trials. I think the first time you see a time machine is probably I Spy, where you're starting a platform. It's 2010. Why don't you talk about the start of I Spy and how a time machine got into that?
Yeah, I Spy 2 is a platform trial in neoadjuvant breast cancer. And just very briefly, it was doing personalized medicine in largely, I'll refer to it as 4, but it's actually 8, 4 types of breast cancer, stratified by hormone receptor status and HER2 status. So you can be positive and negative, positive, negative. Additionally, there's MAMA print, positive, negative, so there's really 8. But let's talk about 8. And what happened in the middle of that trial is that different therapies work differently in those 8 subtypes. And the standard of care had changed in one of the subtypes. And as that platform trial with different arms at different times in that trial, so one experimental arm might be there in the first 2 years, and then another one is there in year 2 and 3. Sorry, 1 and 2, and then the other one's there in 2 and 3, another one 3 and 4, 4 and 5. So multiple investigational arms are in I-SPY, and the standard of care was changing. And the beautiful thing about it was the standard of care being introduced was an arm that was in as an experimental arm in I-SPY 2. And so now we wanted to make comparisons to a new standard of care in, say, year 6 of this platform trial with, at this point, maybe 15 arms. And this is like players in what we'll call it golf, players in golf that had been playing in this common arena at the same time with overlapping eras in the trial. And now we have a new standard of care. And Don and the design team said, how are we going to do this and make these estimates also to the old standard of care? It became important to also do relative comparisons to the old standard of care, but the new standard of care. And this problem lays out exactly like bridging eras in sports, where different experimental arms in a platform trial, in this platform trial, were there. And we were interested in making comparison to previous arms and current arms, actually all the arms, in an apples to apples way, but they were in the trial at different times, and we needed to be able to make those comparisons.
So you got a couple differences here. You mentioned age. You don't have you don't have players getting older or younger per se. Maybe they changed a little bit because of standard of care evolving or whatever that may be, but it's a lot less. But you also have a lot fewer arms in the trial. So you have thousands upon thousands of players in these databases. What do you think is what was the magic number that said to do this?
Yeah, that's right. At any time in I-SPY 2, there may have been 2 or 3 arms over there. And the nice thing is, in many of these subtypes of women, there was a constant control arm. It's almost, call it Hank Aaron. Hank Aaron played for almost 25 years, so he had this really long career. The control arm in that trial overlapped a lot, and there was always a connection. So there was always, every particular point, you could always connect it from a particular arm. So you need at least this connection from one era to the next. If you ever all of a sudden stopped every arm before and brought all new ones in after it, you'd lose that connection. So you need at least one connection in them. And the more connection you have, the more overlap of these bridging, certainly your better ability to understand each era in the trial.
So this is, I've seen a lot of papers consider this. They bring it back to meta-analysis, where connected is actually a phrase in meta-analysis, how many arms overlap between trials. This is essentially the same idea. It's just instead of individual trials, you've got what happened in this trial in 2017, 2016, 2015. Those are what need to be connected.
Yeah, that's right. And so I-SPY 2, just to set this up, so we instituted the time machine, and I think it was the first use of the time machine in a platform trial. And that trial had run over 10 years, 25-ish arms. It's had a new evolution where actually some endpoints have changed and all of that, where we can talk about ways in which we wouldn't want to do this, but over the time, maybe 25 arms. And it estimated the relative comparison of these arms and the effects of era incredibly well over those 10 years. And it brought huge value to that trial. because we didn't have to over-enroll the control. Because of this overlap, we could enroll 1 out of 5 patients on the control, and it was estimated incredibly well. So with less resources, we had great information on the control. And by the way, if you gave over the course of I-SPY 2, what was the estimate of the era effects? That's actually fascinating in the paper of how do the eras change home run rates, batting average rates, scoring rates, going back and look at. In that trial, it was very similar over the 10 years. If you gave the same treatment 7 years apart to the same woman with the same subtype of cancer, you had very similar outcomes. So there wasn't much era effect in that trial.
So Hank Aaron's always 25 and 1930 looks like 1970.
Yep. And in part, it was the incredible aspect that in breast cancer, they had identified these 4 really important subtypes that affected outcome and affected treatment effects. So they had it so well characterized that there wasn't much effect. You could imagine other diseases where we don't really understand that very well, and it may affect some and others, and you may get more era effects.
And I think this brings out one of the key assumptions in the time machine, and it compares it to using historical borrowing, for example, from other trials. If you're bringing in data that's completely, there's none of these bridges, you need that external data to look in absolute magnitude equal to what's currently enrolling. The assumption here is really that the relative differences are stable over time, which would really be in terms of the treatment effect. The differences between arms are stable, but generally you talk about a dead ball era and a live ball era and so on.
Yep.
That's adjusted. And here it would be essentially the same drugs work well in 2013 and 2016. It's not that drugs turn on and off, so to speak.
Yeah, so I think let's get into that.
Yeah.
So I think people can see now we've instituted the time machine in multiple platform trials now that we're using this. And by the way, it's a new thing in clinical trial science with the advent of platform trials that we actually have this incredible opportunity in the same trial to have this ongoing set of data where we have overlapping treatment arms where we can build this and provide better estimates of treatment effect. So in sports and in this setting, it makes this assumption that we're estimating the relative treatment effect between arms. And we can call this, in many of the models we have, we can call this an additive effect. Is that fair to refer to it that way? Now, this could be additive on a logistic regression scale or on a time to event scale, a quantitative scale, but that the relative effect of 2 treatments is constant in any era. Now, if you have that assumption, this model works incredibly well. And there are multiple papers on this. The Roig paper, I don't know if I'm saying her name, Marta. I know Marta, but Roig, that paper that you were involved in that shows you get unbiased estimates as long as you have this additive effect of treatment over there. So while we're using controls from different times, using this adjustment, we get unbiased estimates. Mean squared error is tremendously better. Really nice paper showing this. We have a paper, the Bayesian time machine, Seville et al., that walks through this. So that's awesome. Now, how might this break down? And in sports, I'll let you talk about it in clinical trials, but in sports, this works really well. What might be an interaction? And when people saw this paper, everybody wants to argue that you don't incorporate this or that, but suppose Babe Ruth in the 1920s is really good when pitchers throw a lot of off-speed pitches. Historically, pitchers then threw 9 full innings. There weren't much in the way of relievers. And so he's facing guys that are throwing more off speed. And if he faced guys that threw a lot of fastballs, he might not be very good. And we have an advent now of a lot of fastballs, relievers coming in every inning, throwing a lot of fastballs, and maybe he wasn't a good fastball hitter. So the relative difference from him to Aaron Judge might be different in the 20s, the 1920s, than the 2020s. 100-year time lapse here. Now, you can create interactions like that where the additive effect doesn't hold. You can do that in golf with equipment and all that. But largely, for example, in golf, I don't believe there's any interactions. You can dream them up, but they're not real. And a great player relative, if somebody was better in the 1950s, they would be better now if you move them and they had to play with that equipment and all of that. Same largely in baseball. I don't believe those interactions. Now, what does it mean in a platform trial, Kert?
So for a clinical trial, generally speaking, your treatment effect, it depends on who you're studying and what the disease looks like. So the prototypical worry is COVID, where you've got multiple different there are variants of the disease. And in addition, if you talk about 2021, 2020, you've got lots of severe people in the hospital. Maybe 2022, there are more beds, more people making the hospital that otherwise would have been sent home. You're treating different people. And the idea is that you could have a drug that, because you're treating different people, it has a different treatment effect. In 2020, it works well on severe disease. But in 2022, you're not treating severe disease anymore, and the treatment effect. Now, just like there's lots of stuff on observational data, causal inference, when we talk about that assumption on the treatment effect, you can adjust for covariance. It's after those adjustments. So you can mitigate this risk, but that's the concern, and it certainly would break this down.
Yeah, so infectious disease is one scenario where it might even be a different disease. So an antiviral that attacks COVID in 2020 might be different than an antiviral in 2023. But let's think about ALS, Duchenne muscular dystrophy, breast cancer, lung cancer, Alzheimer's disease, obesity, almost all of these other diseases. This would be very rare that treatment A is better in 2015 than B, but it's worse in 2025 than B. You almost have to try to be creative to even create scenarios where that is. Maybe there's a different background therapy with an interaction to it. But by the way, we have that information. We can look at that. So that could be the potential. A couple parts to this is the important thing. You and I are both shocked by the reaction to this, and we'll come back to that. But you brought up external data. So there's lots of talk in the world now of real-world evidence, use of historical information in trials, where we might run a clinical trial in breast cancer, and we might want to bring in information on previous controls, or in a trial in ALS and previous controls. And this is somewhat all the rage that people are trying to do. That external data, and you brought that up, it's from a different protocol. Patients meet different inclusion-exclusion criteria. The data fidelity is different. The data collection is different to this. It was a different time. All of those things are different using external data. In a platform trial, these patients are being collected in the same trial, the same protocol, the same data. They meet the same inclusion-exclusion criteria. They're in the same trial. They were all randomized. So we have this phenomenal, we can call it historical data. I think it demeans it in a way that is inappropriate. It's the greatest historical data ever to have in the same clinical trial controls. The only difference is time in which they're enrolled. And we can model that.
It's a testable assumption because we've got all of this over time. We can do the treatment effects look different era to era. We also, you mentioned that iSpy changed the control arm, but a lot of platforms don't. You've got this very stable arm that's in there as an anchor to the model the whole time. Certainly these things can happen, but you're at far less risk doing a time machine than any other kind of borrowing or real-world evidence. Now, we should be upfront here. This is not a randomized comparison. So there's a reason that people have this reaction. The magic of a randomized clinical trial is you have at any given time a control group that's comparable to a treatment group. We're talking about bringing in other information. So that is going to break that assumption. The question is, just like anything else we borrow data for, do we get a better answer? Do we treat patients better by using the information as opposed to ignoring it?
We haven't really explained one thing, by the way. We should, and maybe I'll throw this to you. How do you model time? So this question now in a trial, suppose you've got a platform trial that runs for 15 years and you've got multiple overlapping arms. And let's just say we model a year effect. You could model each year effect independently, or you could smooth over time. So our Bayesian model is a little bit different. So describe those two approaches.
So essentially, as you were saying, we bin time. So we could do every year, every 6 months, every 3 months. Statistically, the right thing to do is every time you change allocation, a new drug coming in or not, that should be an error. But a lot of times we approximate this by a number of months and just bin set time intervals. We often call a time categorical model where we just do a fixed, it's almost like we blocked on time and we're setting a separate effect. The time machine, it's got exactly the same structure, but we smooth those estimates. And I think you and I tend to sit on slightly different extremes of this continuum. I tend to do more of the categorical approach. You tend to do more of the smoothing. I think this is like dose response modeling. If you have lots of doses, it makes sense to smooth. If you have a few intervals, maybe it doesn't help as much. But that's the basic difference between it. But basically, you're blocking over time. And then the question is smooth or not.
Yeah, and so that particular problem is largely a time series problem. We're modeling this covariate over time. And how do you model that individually? And within that, and the Bayesian time machine is generally that it smooths. It tends to use a Bayesian smoothing spline to do that smoothing over time. And a lot of the trials I'm involved in maybe has small intervals. And there's not huge sample size in each interval, and hence there's benefits to the smoothing. Where if you have big chunks with lots of patients, their time categorical is largely the same thing. Okay, so it's interesting in this setting. We can even talk about frequentist things like unbiased estimate of the relative comparison of any particular arms and all of this, as long as we don't have this interaction effect kind of thing. Now, people can present examples where if there's an interaction, you run into trouble with this estimate, which is a fantastic thing for us to think about because nobody's ever talked about this before. Maybe in infectious disease, that there's an interaction. And if there's an interaction, all of the stuff we've been doing previously is hugely problematic.
I tend to be a troll when I'm reviewing papers like this. And for what it's worth, I'm often reviewer too, but I'm usually a nice reviewer too, at least. But I usually end up, there's a notion that, oh, this doesn't work and this could happen. And you have these examples. But I often go back to the protocols that have been written and they repeatedly refer to the treatment effect, the treatment effect, the treatment effect. So it's something that, as you said, it's not considered. And the breakdown is actually pretty bad because if you don't think a drug does the same thing in 2022 as in 2023 as in 2024, how do I approve it in 2025 and use it in 2026 and feel confident? Obviously, there's a leap of faith there. I think we all know that's problematic. This isn't a new issue that people have suddenly gone, oh, it's terrible here and it doesn't exist in the other place.
Yeah, and if you believe that this interaction exists, it means that it's hard to ever approve a drug going forward. We need to constantly be testing it in the year to say, does this drug work? Which the cases of this would be frighteningly rare, but it's almost as though statisticians are really good at pointing out this can happen. And all of a sudden, because we're now doing this, is maybe a new problem and pointing it out. So you and I have both been really surprised by the pushback of this. And for last week, we got comments from the FDA. By the way, I'm a huge fan of the FDA, but they were questioning the use of this in an oncology setting in a phase 2 trial of the potential use of the time machine for this. And it's so weird to me that this is a concern in that setting. Of course, within a therapeutic area where historically we have this, where we create objective performance number based on historical data of a 15% response rate that you're trying to beat. And now we're trying to use this time machine that there's this almost this reticence to use it, which means you ignore a ton of data from the same trial. And it just, that has surprised me greatly that there's been a pushback to this.
And maybe I'll put back a little bit on one thing you said. I do think some of these interactions could happen. You could imagine a treatment effect. It's varying from 20 to 25% over the course of the trial, some unmeasured covariate. Usually the magnitude, people worry about type 1 error inflation in this. If you're talking about small differences, you get small type 1 error inflation. It's 2.6, 2.7, 2.8. A log rank test will do that. Doing basically most asymptotic results do that. We do this all the time. So I accept the possibility, but I don't accept that it's a catastrophe.
Yeah. Yep. And ignoring really valuable data has huge impacts, much more than the 2.6 that you talk about.
Type 2 errors matter too.
Yeah. And the other implication to it is you collect a whole lot of new control data that you probably don't need to collect by using the data you have in the trial in really smart, particular ways.
And certainly I would recommend ever eliminating a control arm completely. That's not what we're talking about doing.
Yeah, and that's the huge value of being able to use time, is having this overlap. And a control arm is a huge value to that. Now, I don't use the time machine in some settings. There are some diseases, like I was involved in a number of COVID efforts, and the REMAP CAP trial, for example, does not use this non-concurrent controls. And it was the disease setting, it was thought not to do it. Now, we use time adjustment, even though we don't use non-concurrent controls, which actually confused some people thinking we were using non-concurrent controls in this setting, but disease specific. And there may be other diseases where there's massive changes in the disease and in the way it's treated that we worry about exactly those things. So this can very much be disease specific. And then there are other diseases we know largely there have been very little differences over time.
There are some cases where you never know what happened. I remember when we were involved in Los Angeles modeling of COVID, and there were about 3 times during those 18 months, whatever it was exactly, where clearly the amount of time people spent in the hospital, it changed. We don't know why. We had to address it in our modeling. These are the kind of things you can't anticipate.
Yeah. Now, as it's also one of the huge values of these platform trials is actually this estimate of time. It becomes a scientific quantity of interest. And I brought this up in the sports example, that looking at Major League Baseball from 1920 to 2020 and the relative, what a player would have done if they played at the different eras, those estimate of that is fantastic stuff for that. And looking at the NHL, for example, in the mid 1980s, a player would have scored many more points by playing there than they would play now. It's just a different sort of game. And that fantastic thing. The same disease learning happens in these platform trials with these estimates of time. And we have a number of these trials showing this. And as you say, we can explore, is there any evidence of interaction within there, if that's the thing that people are concerned about in these trials?
I think one thing we ought to say here is, I think people get reasonably nervous about the thought of talking about I Spy borrowing data from 2010 to a drug that was enrolled in 2020. So the notion of what does that really mean? Are you pooling that? So on. And certainly this isn't what that model does. It depends on the overlap. So when you're doing sports, you've got thousands of players going back in time. In a clinical trial, we don't have that. We maybe have a couple dozen arms. If you're looking at a drug in 2020, you give some weight to the patients that were enrolled in 2019. You give less weight to 2018, less weight to 2017. And it depends on this overlap and how many patients. We're not pooling patients from 2010. They're largely discounted because of the lack of overlap in a clinical trial.
Yeah, that's a beautiful thing. And I think you did this in the Roig paper et al., where you look at, in a time categorical, you can actually calculate the contribution of a patient depending on the overlap from earlier. And it's this beautiful, the variance covariance matrix gives you this beautiful weighting of the patients, and you get this natural down weighting. It's a beautiful thing.
And the Healy trial uses this to some extent because there's a window. It doesn't borrow back in time and infinitely. It borrows back to a certain degree.
Yeah. And you're a fan of potentially even just algorithmically cutting a line and saying, we're going to do this for the last 5 years or something. And that might be disease specific as well, depending on what we think about how the disease, how the treatment has moved within these trials.
I think you could just basically look and say, this is only contributing 1% of the effective sample size. This isn't worth it. You avoid problems with people having to publish and there's live data that's still being used. A lot of problems go away if you eventually realize this really isn't helping us much. Let's put a line in the sand and cut it.
Yep. All right, Kert. So if you could use this time machine and go back and tell Kert Viele at Carnegie Mellon, as a graduate student, you could go talk to yourself, what would you tell yourself?
Oh, that's a terrible question to ask. Let's see. So I remember when I was in grad school, probably the least likely thing that you would have ever thought I would do was end up as a biostatistician, because I was incredibly computational, incredibly theoretical. You'll remember we had this bet on who could use you wanted to use the fewest theorems in your dissertation as possible. So whereas I had pretty much ruined that already. So I think this notion of the applied stuff is where you can actually really affect the world would be on that list.
Yeah. Awesome. I did have a theorem in my dissertation, by the way.
Did you? You had one.
Yeah, I think I had a couple. Yeah.
Okay.
We need to do one of these on my dissertation at some point, which for those interested, it was how to optimally play hide and seek, which you may find interesting. By the way, if I could go back in time, I might tell myself to do a different dissertation topic, but I enjoyed it. I loved it, and we're in a good place.
I have never revisited my dissertation.
Yeah. We're in a good place. You know where we are, Kert? We are in the interim. And till next time, thank you all for joining us here in the interim. Cheers.